Hi there.
Welcome to Microbiology Journal Club, where we nerd out big about all things small.
My name is Danny, and in a previous life I dropped out for my PhD in microbiology. Then I was a fact checker and editor for pharmaceutical advertisers, and nowadays I'm a member and the president of Biotech Without Borders, a nonprofit based in NYC, dedicated to the public with the tools of biotech.
My name is Faz. I have a PhD in microbiology where I mostly worked on bacteria, and since then I've worked as a research integrity specialist. I'm currently working as an editor for a scientific journal.
Every other week we meet to talk about microbiology, and on a typical day like today, we do an overview of some of the coolest microbiology papers that we've seen in the last 3 weeks in this case.
Yeah, and also we want to do deep dives into viewer-selected papers, so leave us a comment or tweet with your suggestions for a deep dive, and we'll look into making that content.
Yep, you can follow along with the papers that we discuss on either week in our shared Zotero library. Our social media handles and the link to the library are in the doobly-doo below.
And boy, do we have a show for you today. First, we start off, as ever, with COVID, with a trial to determine if Molnupiravir can actually treat SARS-CoV-2. Then we look at what may cause long-term smell loss in COVID. And finally, a test that can potentially detect all viral infections, even the ones we don't know about. Not only that, we have also found the missing link in the evolution of all multicellular life on the planet, a parasite that causes memory loss in wasps, Virovore, an organism that can live by eating only viruses. Take that, Kaspersky. Also, we get can artificial sweeteners be used as antibiotics? And finally, a yeast bioengineered to be a narc. All that and more, so stay tuned.
Mm-hmm. And so, as Faz said, our first papers are about SARS-CoV-2, and the first one we have on the docket is Molnupiravir plus usual care versus usual care alone as early treatment for adults with COVID-19 at increased risk of adverse outcomes, an open label, platform adaptive, randomized controlled style trial.
Yeah, so Molnupiravir is like, we discussed this on the channel before, but it's like one of those broad spectrum antivirals that mimics a nucleotide and it's almost designed to directly mess with the SARS-CoV-2 polymerase.
Mm-hmm.
So they've got a RNA-dependent polymerase that they use that is found in SARS-CoV-2, but not in like us. So if we target to that enzyme, then that means that we don't get affected with bad side effects. And I think the main thing that people were very excited about was it was an oral antiviral. So unlike remdesivir, where you have to like kind of inject that directly into the veins, having a pill that people can just take is much more convenient, and I think much more practical. I think for widespread use. So there was a lot of like kind of people very excited about Molnupiravir. I think myself included.
And
I think
I was gonna say, I think like because it is a nucleoside analog, there's that same idea like, well, what's the effective window for it? Because like it has to be present during the time the virus is replicating, just like remdesivir is. So I guess that's probably something that like has to be watched out for in terms of whether it's going to be a really good tool.
Yeah, I agree. I think the first time we covered this, it was like a press release, but I think the key thing that we noticed was that press release was the like study describing that was very targeted towards that very early stage of viral infection. So I think within the first few days, and that, I think we've talked about this for other antivirals, where like those first few days are very crucial for antivirals to work potentially. Well, at least that's a theory that I've got going.
Yeah. So I mean, that's just I think when people do these studies, they have to be really aware of like that window. And I think specifically also in this study, like this is like it's a really big study. I think it's because Molnupiravir is already approved for use in something else. That's why it's open label here, and they're just like as many people as they can enroll in it that become unwell. They get urgently sent a packet of this drug.
Yeah, the entry requirements for this is you have to have had like SARS-CoV-2 within the last 5 days, and then they they mail you a package. Of it, and then you take it day on day. And some people got like other things mailed to them, like things to take blood tests and swabbing tests, so they can get more data about how much virus people have inside of them at the time.
Everyone was 50 years or older, I think, in this study. They said they
were. Yeah, that's right. So again, this is like a vaccinated cohort, because I think it's getting much harder to find an an unvaccinated cohort. Anti-vaccinated cohort, just type in the comments you like vaccines, and then they'll appear.
Yeah.
But yeah, so I think antivirals, because antivirals tend to not do so well when taken into large-scale trials and actual places where people have to practically use them in a practical sense. I think the main success would be PrEP, where people are given antivirals pre-exposure. That's usually when it's successful. And I think, and also in preclinical models, where again, in preclinical models, those tend to target the very early stages of infection, because I think that they bias towards trying to get the best results possible. And so I think,
but I think that's what's nice about this study, is that it's happening in a large amount of people sort of in these real world conditions where they don't have super good control over when exactly they get to give the intervention. And so, yeah, any good result from here is quite, it would be quite positive.
Yeah, that's true. And there is maybe one good result, but with a big caveat that I'll go into.
Yeah.
So they.
Yeah, so I think the primary endpoint, the main result that they're looking for was whether or not it stopped people from going to the hospital. Right? They want to prevent that, which, I mean, that's quite a big one. But I don't think that result came up super significantly.
Oh, no.
Yeah.
They basically found that. So, the full results of the study was that the main endpoints they cared about was death or getting hospitalized, and they found that Molnupiravir didn't seem to have any effect on preventing that.
Right.
That compared to control, there wasn't any differences. The only differences they found was, so they got this one graph that looks very positive until you figure out what it's actually showing.
Figure 3.
I'm referring to because they asked, there are the primary endpoints we talked about. There are also secondary endpoints, such as, hey, how are you feeling once you're having this disease, this treatment? So there's one graph where it seems that the Molnupiravir was having a better. Looking at this graph, it looks like, hey, lots of people are recovering compared to the usual care group. But then you realize that what this is based on is people's self-reports on how well they feel.
Yeah.
And since this is an open label trial, there is no placebo there. So this could be a placebo effect, because there is no placebo control. So the people getting usual care don't think they're on some extra magical drug, whereas people who are on that might tend to feel better because they're being given this extra elite medication. So again, that looks positive, and obviously the thing that we really want with medicines is to make people feel better. But I think it's hard to tell whether it's Molnupiravir that's causing that, or whether it is a general placebo effect.
Yeah.
Another thing that's fairly positive is that they did take swabs from people, and did find that the actual viral loads were lower in people who received Molnupiravir, but that just didn't seem to have that effect. That didn't seem to affect the disease outcomes, which again it's both I think it's one of those things where it is positive, and it does kind of highlight a flaw in how we study these antivirals, because if we were just doing this in animal models, we'd chalk that up as a win, because a lot of animal models use the viral loads as a proxy for how well infection is going. So having Molnupiravir reduce viral loads is basically what people think should be happening at this stage, that less virus equals more likely to survive. But I think this study maybe shows that isn't necessarily true, and there's much more going on there that we need to think about when applying antivirals.
Yeah, I mean, mechanistically, we already talked about the reason why, is that it matters when the viral load is reduced in the course of infection. So, right, probably the viral load is already, in this 5-day window for which they could get the Molnupiravir, maybe the viral load has already spiked up and sort of is on the way down or is stabilized another way. Immune cells are already doing something. Events have been set into motion already, and being able to reduce the viral load at that moment isn't helping. I mean, again, that's just a potential theoretical way in which, it wouldn't work as well.
Yeah, I mean, the thing that I tend to think about is in terms of buying time for the immune system, so that having the antiviral early along buys time for the immune system to develop a response against the virus at a point where it's likely to change the course of infection. But once it's like gone, again, this is all like out of my own head, and it's not like peer reviewed at all. But this is like my idea, my conceptualization of what I think is going on.
Right.
That there might be some point where the disease has progressed too far, and that at some point the antivirals aren't going to have that value added for the immune system, that say like compared to vaccines and other things where the immune system is like prepped in advance.
And I guess that's the interesting thing, is that like from us covering these types of papers, these clinical papers, is that like we get to make up these funny, interesting sounding mechanisms on what we think might be happening. But at the end of the day, like we have the data to show that like maybe it doesn't have the clinical impact, and you know the clinical impact is what's important in terms of actually saying like is there evidence to use this particular medicine in this particular way.
Yeah, if anything, this has kind of undermined my like faith in whether any antiviral is going to be extremely effective on its own. Because I think what this has kind of highlighted for me is like there is this like gap in the pipeline between like what we look for in our preclinical models and what we look for in clinical models, and how to address that is going to be very, very difficult.
Yeah, that's. I think that's the all those when we were going through grad school, I think this translational research term got thrown around a lot, and it's a huge gap actually between the clinical models and the lab-based models.
Yeah,
but yeah.
My entire master's was about like how the difficulties of like translating from animal models to post-clinical models. So maybe that is just my bias showing. And also, like, again, like we're going based on mechanisms, but we don't like necessarily going on with these antivirals. And the thing that is important to people is whether they work or not. I think that, and saying that they should be applied earlier, I feel like is playing into the hands of the people making these antivirals, because then they can say, oh, just use PrEP and all, or like, almost feel like I'm apologizing for this antiviral not working rather than trying to. Well, I'm just following my curiosity, but I do realize that this could be like a dummy. It could be like something that doesn't work, and I'm just like, it could work because of blah blah blah, and yeah.
Right. Just let's all have every prophylactic.
Yeah, and I feel like that what I'm saying is that based as a researcher, but if I was a doctor, I'd probably punch myself in the face.
Yeah, there's different levels of criteria. Yeah, I think that the concerns are really different in these different disciplinary domains about what's important and what isn't. But yeah, anyways, I think it's great that they brought together this data and published it, because it's important to know how things work in the quote-unquote real world, even though there's like all sorts of other caveats there.
Oh yeah, papers like this that show the real results and show real negative results are, I think, really, really important, and they don't get published enough. Yeah, I stand negative data. Like, it's also because people don't talk about that bias that all researchers have. With like, I'm working on this drug, I really want it to work because it'll be good for my career. And I do like trust it when a lot more when researchers bold enough to go say, hey, we tried this, it didn't work. I'm like, yeah, I believe you, because it takes a lot of bravery to go out and say that.
Totally. All right, well, let's move on. The next thing that we have is persistent post-COVID-19 smell loss is associated with immune cell infiltration and altered gene expression in olfactory epithelium.
So I think this is another, like, following on from like the translational model thing that we're talking about, because we have looked at this kind of question, but in hamsters, where they did some quite interesting research showing how COVID-19 affects the olfactory epithelium in hamsters.
But yeah, I think in hamsters we had seen that it can potentially get into the neurons and stuff and mess stuff up. But that was in hamsters. But then I was reading the introduction of this particular paper. They're saying in a lot of human autopsies, they don't see it specifically in the neurons of the nasal epithelium. And we had covered one of the papers that they talked about in the introduction previously as well, which was like a 23andMe genetic polymorphism one, where they in a data set from that company, they were able to narrow down like some particular gene set that correlated with, or like that was affected some way by the SARS-CoV-2 infection, and like that was not related to neurons either. It was like an epithelial gene marker of some sort.
Yeah, so this actually looks at like real tissues from people. I think these were like extracted during operations that were related to, I think, things that. So they're looking at healthy people who were getting something that wasn't related to a respiratory infection. So that, because one thing you'd be concerned about is if, because this is looking at a very late stage. This is looking at post-acute COVID-19, right?
Yeah. So it's also people who are reporting. Yeah. So one set of people like are just healthy, getting some sort of routine nose thing done, and then another set of people who have said that they had previous COVID-19 and now they can't smell anymore. And that's like continued. So.
Yeah, this is part of the whole long COVID research where like there are persistent symptoms long after the COVID-19 is gone. And I think that's confirmed because they do a lot of tests to see whether the virus is still there, and they find that it comes up negative, but there are still people without smell. And they take samples of the olfactory mucosa, and they find that things like there's less neurons around, there's T cells aren't in the places you expect for a healthy mucosa. I think there's a lot about the gamma delta T cell response in this.
In some ways, it's a big immune cell. They're using all this single cell where they have to, where they're able to collect the expression profiles of all the different cells that they have in their biological samples, and then reduce the dimensionality of that and cluster everything, so you get these really great. I mean, some of them are really distinct clusters of cells. And that's because they have specific expression profiles that are unique to them and them alone. And the sort of, we briefly talked about this before, we won't go into detail, but like, through the magic of computers, they're able to give us, be able to distinguish these different cell types from each other. And so then you can say like, inside of a disease state versus a non-disease state, which cell types are going up or going down.
Single cell RNA sequence is such a great technology for pulling out some interesting potential hypotheses. So I almost imagine it's like a net trawler that's trying out these hypotheses that we can test in more detail later, but it gives us a broad idea of what's going on in these in these particular patients.
And so applying that tool into the sort of the best model, aka the actual human people who are experiencing these things, gives us the very relevant data that then again you can backtrack that into an animal model later on to confirm hypotheses.
They make also specific mention of sustentacular cells, which are a cell type that I've
either.
But they're really important for the olfactory epithelium because they detoxify harmful chemicals, and they do a lot of like important, I think, protection stuff in terms of modulating mucus layer of the olfactory layer. And apparently
if you scroll down to Figure 4, that's the year they talk about these sustentacular cells, and they go into the gene expression changes that are happening in them, and also come to this hypothesis, right, that immune signature that they saw that's getting upregulated, that's partially what might be causing this dysregulation in these other cell types in that epithelial layer.
Yeah, I think this is like interesting, not just for the idea of what's happening specifically in olfactory mucosa, but also like getting an idea of what the immune system is doing during post-acute SARS-CoV-2 sequelae. I think that getting an idea of why the immune system is still like kind of got switched on after all this time, I think that's going to be quite important to figure out.
I didn't really. Personally, I had not heard of the fact that some people still don't have their sense of smell after SARS-CoV-2 infection, and so, it's horrible. But like this is one of those examples of natural experiments where, in a subset of individuals where this fate has befallen them, they're able to get sort of a scientific analysis of like why does it happen in this particular subset of people, gives us insight into stuff about the immune system that again might just be, it could be generalizable even in terms of the immune lessons, right? Because I think a lot of the post-acute sequelae, it's thought to be something about the immune system, but how do you begin to ask the question, what is that something? you need sort of a test cases, and the loss of smell is something that people notice acutely, and so they actually, it serves as like a really nice, and you've seen you can measure it as well. There's also tests for smell. Can you smell this or can you not? Yeah, so it serves as a really useful test group to be able to learn some of these lessons about the immune system in relation to COVID-19 and post-acute sequelae.
Yeah, and I think we're going to move on to the next paper, which is Nasal Host Response-Based Screening for Undiagnosed Respiratory Viruses, a Pathogen Surveillance and Detection Study. So the COVID-19 has like elicited this fear of unknown viruses, which I think is justified, but that's because I run microbiology YouTube channel and we directly profit off of this. So I don't think we should necessarily take that on to heart.
We're not directly. We're not profiting.
We're not profiting money-wise. We're profiting by like this
attention,
I guess. Yeah, I don't know. We're not profiting at all, but it feels weird.
Just want to make it clear to people that we're not like raking in the dough with our 400 subscribers.
We're not raking any dough at all. I just want to make it clear that it feels weird to be happy about that. That's that I don't feel good about being
happy that virus. That people are afraid of viruses.
Yeah, that's not someone who really is interested in microbiology. Seeing that this other group of people now interested in it because of the fear, it doesn't feel good.
Yeah.
But yeah, anyway, so this is because this study is about trying to find undiagnosed respiratory viruses, new viruses out, because I think people are very aware that could be quite an important thing to know for the future. SARS-CoV-2 started out as an undiagnosed respiratory virus. So having a screening process to be able to hunt them out would be quite useful. So they've come up with a strategy of well, because at the moment most tests are based on we know what the virus looks like, we know what to look for specifically, but if you don't know what virus you're looking for, then you don't have any of those tests available. So that would just turn up negative, so you wouldn't see it. But our immune system is very good at spotting like unknown viruses. It's designed specifically to hunt out viruses it's never seen.
And I think specifically too, the innate immune system branch. Like we talk about the adaptive immune system where all the antibodies come from, right? That requires having seen something before, right? That's how vaccines are great because they train our adaptive. But there's this other arm of the immune system we call the innate immune system, and that is this long genetically encoded system of different sensors, and that system has to be flexible to sort of throw up the early alarm to even begin to distinguish infection from non-infection. And so that's the system that these authors are trying to exploit in a way to see like what signal could come from the innate immune system that has been tuned through evolution to be able to detect this sort of thing that then we can piggyback off of and be like, that means viral infection.
Yeah, and the signal they latched onto was CXCL10, which is one of the signals produced by the innate immune system when it encounters viruses. So that's kind of great because I guess there's a cynical thing where you could say, okay, but a runny nose could also be a symptom of viral infection. But I think that the way they apply it here is quite interesting because this isn't like they're not looking at individual patients; they're looking at like whole health systems where they'd get nasal samples from people and they test them in a massive batch to see whether the virus is in there. And what they did was they took like some historical cohorts of virus samples taken from before the outbreak and like in the beginning of the outbreak where there weren't any COVID tests. And they tested them for CXCL10. And in the first cohort, I think they found like some viruses they wouldn't expect to see, like influenza C, and like seasonal coronaviruses in tests that turned out negative. And for like the ones when the coronavirus was just advancing, they found that they could find coronavirus in that this test is good for detecting whether there is an antiviral response, and therefore can be quite good for narrowing down and finding out what viruses aren't being covered so far in
and CXCL10 is I think an interferon stimulated gene, and we've talked about that a little bit before on the show. Like that's a very common pathway, right, of antiviral response in our cells. So I think like Epstein-Barr came up too sometimes. Like some viruses you wouldn't expect to cause some sort of respond, like have at least symptomatic sort of manifestations, but they're inside of this biobank, and when they tested them for CXCL10, it came up positive, and some viruses were there. So yeah, really interesting technique. We were saying earlier, like some people might say, well, a runny nose is also our body's immune response, but there's a convolution there, right, where it's like it could also be allergies, right? It could also be are some other things that cause runny noses that aren't just viral infections. So I think the way that the study was set up is they kind of went up the chain a little bit from that symptom, right? Try to go closer to the source of just exactly what is the immune response to viruses.
And to be fair, there is some utility in like antibiotic stewardship, where like being able to know whether a disease is viral or bacterial can be very, very helpful. Because I remember like back when I was a kid, like my doctor would give me antibiotics for pretty much any cough or sniffle, and then they realized no, if you do and so I think that this could be quite helpful for people who, if it's encapsulated in a useful test, it could be quite helpful for people.
Yeah, absolutely. I don't know. Let's show a figure, at least one figure from this paper.
Yeah. Oh, sorry. I'll scroll down. Apologies.
I thought figure 4 maybe was like something kind of nice, just because like they have a clear set of negative controls. Then they had the SARS-CoV-2 peak is in like the red, and then they have rhinovirus, and like so in this particular one, panel B, like the rhinovirus, like some of them come up, but some of them don't come up. So like it does seem like this signature is like specifically for SARS-CoV, like is more enriched for SARS-CoV-2 than anything. But yeah, also other viral infections do sometimes show up with this particular test, at least based on the data set that they have.
Yeah, that is an important point, because they could pick, there are lots of different cytokines they could choose from, and the signature might be different depending on the kind of virus that's coming to it.
Yeah, so. Absolutely. Yeah, so like in this panel, like it looks like Trail also is like a cytokine that CoV-2, but then like in some other cytokines they try, like CCL22. That one is like 50-50 showing up SARS-CoV-2 or not. And I mean, this just we always talk about how complicated the immune system is, so like it should be no surprise to our listeners or anybody who knows stuff about the immune system.
Yeah,
trying to find like the one thing that the immune system does the same in response to something else, like that's not always a straightforward task.
Yeah, and because like in the patient cohort, you're catching the immune system at different stages of an infection, and it's not always going to be the same what kind of cytokine signature you are. I mean, there's no given like that the immune system will pump out the same cytokine throughout the entire course of an infection.
Right. But I think the hope is that maybe, like, their hope with CCL10, just based on seeing the results, is that it is something that is somewhat constant during the infection, at least when it comes to SARS-CoV-2.
Yeah, I think so. I can't remember what CXCL10 does, but I think that... I
actually have it up on Wikipedia, but I don't know. It just says it's a cytokine. There's something immediately that says function. It's in response to interferon gamma. It does things to cells, macrophages, T cells, promotion of T cell adhesion to endothelial cells. Who knows what it really does? Because they have multiple roles, also depending on cell context. So,
yeah, it's kind of like a generic messaging molecule that can have different impacts depending on what cell it interacts with and what context it is, I guess.
And I mean, it does have, like, to a more experienced immunologist, they could probably tell us, right, exactly like what it tends to do versus what it tends not to do. But I can't get that just from skipping this Wikipedia article. All right, let's move on.
Yeah, this is an exciting paper on. I'll scroll up this time. Actin, cytoskeleton, and complex cell architecture in Archaeon.
Mm-hmm. So, Fuzz, you introduced this as a missing link type of paper, which I think is great, because if people know about this like big phylogenetic tree of life, and then some time ago they realized, oh wait, there's another group of tiny single cellular prokaryotic organisms that exist but when you do their phylogeny, they're actually more similar to eukaryotes than they are to bacteria. And we call this group the Archaea. And so it's like, oh my God, this like another set of microscopic life that exists out there that's more closely related to us than bacteria. But there were still a bunch of, I guess, proteins that were considered specific to eukaryotes. So in this paper, they're calling them eukaryotic signature proteins, or ESPs. And, you know, we consider that like our innovation, right? Just for us, like no Archaea have these things. But the big discovery in this particular paper is finding some structures that kind of look like structures you would find in eukaryotes, and then finding out that those structures are produced by proteins that are analogous to proteins in this signature protein suite. And so, yeah, where they started, Fuzz had us on this like enrichment procedure at first. So the Archaea that they're looking at are from this group called Loki Archaea, coming from sediments. And they specifically use like an easy to access sediment. So one of the contributions of this paper is just this culturing technique of enriching for this particular type of Archaea inside of an enrichment culture. I think they talked about using like a minimal medium was really important, which happens a lot in microbiology. Like there's so many microbes out there, they don't all want to grow when everything is given to them. Some of them are waiting for their opportunity to grow, which means a very specific set of conditions where they think they'll be more successful if they start dividing rather than something else.
Yeah, these kinds of bacteria are very finicky. They're very fastidious, where like and half of this paper is actually trying to figure out how to make this Asgard Archaea grow, and it's because you're effectively capturing a wild wild organism from the environment and trying to figure out how to keep it alive in a lab, and that especially for like Archaea like this, because Archaea, they don't live alone. They are in symbiosis with other like organisms that like desolve real methogens that kind of it shares like nutrients with. So it does syntrophy, which means that you can't necessarily grow these things alone. And in order to make sure that you have to keep its partners alive, you have to be able to really like do a lot of like legwork to make sure that you know what you're growing and it's still alive at the end of the day. And
I mean, I think a nice way to think about this too is that we don't know what they eat. Right, and the syntrophy makes that particularly complicated because they might eat something that something else makes, and so like, how can we capture all these molecules that could be potentially food for the organism and make sure that we're feeding that one but not other ones? And so, yeah, it takes a lot of testing, a lot of trial and error to come to a set of conditions. And they talk about a little bit in the paper, like they tried a whole bunch of things and not everything worked, but they were able to find something that got the percentage of Loki Archaeota to like 80% or something like that. Yeah, they were able to get up to 80% after like a set of transfers. So the whole culture is not Loki Archaea, but it's like a large majority, and that's really helpful in terms of doing any sort of experimental procedure now. And so the procedure that they decided to do was they did some genetic analysis by looking at the genomes, and then they also did some microscopy.
Yeah, cryo-electron tomography. Well, they did some microscopy as well. So they did some fish. But the problem with fish is these archaea are very fragile, and fish just pokes a lot of holes in them, and they're concerned about destroying the structure of the cells.
Yeah.
So they had to look at them under Cryoem, but Cryoem, they'll see both like the Archaea and the Symbiosis as well. So they have to figure out they have to figure out how to tell the difference between them. And they'd usually do fish to like kind of co-register them. So like, oh, this RNA sequence is found in this bacteria, and we've got a little probe that'll bind to that, and it'll send out a light. And then we can tell that this organism is definitely the Archaea we're looking for. They couldn't do that but they did a really, really interesting strategy.
Yeah.
Do you want to talk about that?
Sure. So I guess they know from looking at the genome, right? That the ribosomes of these archaea, like they have an extension, a particular chunk of RNA that sticks out from the ribosome in a way that doesn't stick out on other things. So essentially, these Loki archaea, they have a different shaped ribosome. And because Cryoem is this amazing sort of structural, or a very high resolution way of looking inside of cells, they use the shape of the ribosomes that they're able to reconstruct in the fields to identify which archaea is the one that they want to look at. These blobs, right? Because it's all blobs under the Cryoem slices. And yeah, they're able to narrow it down to find the things that they're looking for. Yeah, I think that was pretty wild.
I think that, yeah, that's amazing. I feel like that's going to be a really important like technique in the future, because especially for looking at mixed cultures and how bacteria might interact with each other, just being able to like have that kind of detective work of being able to identify like bacteria on site based on the structure of their proteins. That's just like,
like yeah. I think it's definitely going to come from like ribosome is such a important, like everything needs a ribosome essentially. All cellular life needs a ribosome. So yeah, being able to capture the ribosome or infer ribosome structure from genomic sequence, and then confirm ribosome structure visually in a microscope, like that, those two things interplayed, as you say, is just going to give us a lot more stuff we can look at.
Yeah, I mean, the very idea of doing that would sound completely insane like 10 years ago. I feel like.
Oh yeah, for sure.
Yeah, just like I remember in my microscopy classes being like, oh yeah, we'll never know like at this level because electrons and blah blah blah, they're not going to be good enough. And then like, wow, we've got this amazing image that it's a lot. It was a lot to like computing and advances in that. So
yeah, absolutely. And also it builds upon the back of all this structural biology of ribosomes, right? Like you have to be able to predict that shape from seeing like a ribosome sequence. And we don't have that ability unless we knew so much about how they're put together. So yeah, anyways, so they're able to look at a bunch. And so in figure 4, they have like both scanning electron microscopy images of these Loki archaea, as well as the Cryoem slices. And they are weird appendages, tentacles coming out. And also they see like sort of these densities that look like filaments of some sort. And this is where the actin, they're thought, like this could be actin cytoskeleton stuff. I mean, I think they had already seen the homologs when they got the genomes. So like this is like now sort of like confirmation that those genes are indeed producing filaments. And if you look at the sort of the middle bottom picture of like all the weird blue things, but there's those yellow strands, those filaments, they're going through the tight choke points. And so the authors are inferring that that gives some evidence that they might be structurally related because they're maybe keeping it open or stabilizing those really long membrane appendages.
Yeah, I think they got a little video that shows how they build up the images, right?
Yeah, let's watch it.
Yeah.
I don't think I saw this.
Yeah. Again, they always hide the videos in the supplementary sections, but it's always good. So again, you see these protrusions in the cell body. They target one particular. So they're taking slices throughout the entire thing, and so they can eventually build up a 3-dimensional image using the Cryo-ET. So I mean, I always love these sorts of things. I'm a sucker for videos, and yeah.
Oh yeah. And the video is just so good at showing, like you can see how the focus is changing. Each one of those, it's actually they're going through slices potentially with each one.
Yeah, and you can see like these orange filaments, and you can see these like gray ribosomes, plus like the inner membrane and the surface, and you can see kind of how they interact with each other, which it's pretty cool to get that kind of detail.
Yeah, and that's all just through sort of natural electron density, and these organisms, they're captured inside of ice. I mean, we've talked about cryoem a bunch now, but just to remind people, it's like this weird form of like vitreous ice that captures the organism. Oh man, yeah, this is wonderful.
Yeah, and like and back in the old days where you would just focus on one slice and that would be all we get. Now you get multiple slices and we can build up these amazing images where you can get.
Yeah, that's robotics there as well,
Yeah.
right? Like back in the transmission electron microscopy days, I think robotics was slowly coming into there and being like, if you could have a robot take perfect slices one after another and grid serially, then you could get it. And before, you would need like a really skilled person to be able to do that, and that was a skill that not everyone could do. It was a rarity. Yeah. So again, because Cryoem has such good resolution, they're even able to have like it's not great. It's 27.9 angstrom structure of this filament.
Oh, only 27 atoms. How? God.
Well, I mean, like from the crystallography perspective, like we're not. Yeah. This is not good enough to figure out where each residue is sitting, but it's good enough to find the overall shape of things. And specifically, the feature that is very nice that they're able to resolve here is the handedness of the helicity. So like they're able to say like, oh, okay, it's a very specific type of helix, and that gives this idea of like, oh, it twists this many units, right? You can kind of like approximate where the molecule might fit inside of this helix structure, and so again, not the most resolved in the structural biology world, but surely enough to really say some very interesting things about the structure of these filaments. And they really do look like actin filaments, and they really are related genomically to actin inside of eukaryotes. So again, this is the missing link element, right? Like we've never seen these proteins before, not in eukaryotes, or seen proteins like this, not in eukaryotes. And so it's like really exciting to find them in Archaea. Maybe not unexpected because we saw that phylogenetically they sat in between us and bacteria. But yeah, this really, this is really quite exciting, and it fuels a hypothesis by the authors about like maybe the reason why there's all these protrusions is because they're trying to get close to their syntrophic buddies, right? Their cross-feeding buddies, and maybe that's like a model of how endosymbiosis could have happened in the beginning, like not taking something in, but rather trying to reach out to something out there.
Yeah, I mean, I think because we did see like electron microscopy of like symbiotes and like their archaea like last time, and we did see like they do like kind of get up in there. There is a big group hug when it comes to.
Oh yeah, they're in little cluster balls usually. Yeah.
Yeah. So I think that the hypothesis of like not that it ate, but like just kind of like hugged it so tight that they became the same.
Yeah.
The ultimate codependency.
Another tool in this figure that they created was they were able to raise antibodies towards this particular Loki actin or whatever, and that's a really useful tool now that you can go out and use is being able to stain for this particular molecule. And yeah, I think that's the big discovery of the paper and those implications in terms of what it means for our evolution. And yeah, I'm blown away. This is just really cool, and also I think it also highlights how much we don't know about Archaea. That like this molecule that's actually pretty prevalent. Once they were able to culture a bunch of it and do a full genomic sequence of one, oh, there's all these weird analogs to eukaryotic proteins. And actually, the introduction. Sorry, part of my excitement about this too is that I didn't know a bunch of the stuff in the introduction. Like they were saying that in the introduction, like escort proteins, so the stuff, the Golgi apparatus, the way that vesicles are made, like those proteins were also thought to be eukaryotic specific, but actually upon sequencing a bunch of Archaea, they also find homologs there. So like there's a lot of stuff that actually is inside of Archaea that are related to eukaryotes, and I just didn't realize that before, and that's what's really blowing me away. Just like talking and thinking about it right now.
I mean, the thing that I'm really excited about is that they've now got a way to grow this Archaea in a way that it can be used in a lab, because that's probably the big thing, having a way to like grow like an Archaea that doesn't take like forever to grow up.
Yep.
And it doesn't, so it doesn't like, because things can go so much faster when you have a bacteria that, sorry, not bacteria, but an organism that that you can actually take less than a month to grow.
Mm-hmm.
I think that
Mm-hmm.
because if you're doing a PhD, it's a bloody nightmare that you have to wait a month to figure out when your experiment goes wrong.
Totally.
I mean, that's yeah. So I think having something like that is just a real game changer for people working in archaea research.
Yeah, yeah. All right. Well, let's move on to the next one.
So, yeah, at the threshold of symbiosis, the genome of Obligately, Endosymbiotic, Candidatus, Nebulobacter, Yamunensis is almost indistinguishable from that of the cultivable strain.
Yep. Yeah, this is a real short report, but I thought it just had a really interesting observation, and they also just trained their eyes on a very interesting circumstance. So a lot of ciliates have endosymbiotes, a lot of protists have endosymbiotes, but particularly ciliates. And in one of these ciliates, Euploites, they have this one endosymbiote that's Nucleobacter Yamunensis, and that bacteria has a free living close relative called, it starts with F.
Fastidiosibacter lacustris.
Yeah. Thank you.
Took me 3 goes to get it, but eventually I'll get it wrong again in the future.
And so it's known to be very closely related by 16S sequencing. So like they have practically identical 16S sequences on the ribosome, but it's known for a fact that you can grow Fastidiobacter on an agar plate, but you cannot grow Nucleobacter on an agar plate. And so these two, the authors of this paper, they culture a whole bunch of it, one inside of the protist, the other on a plate, and then they do a genome sequence on it, and they compare those genomes to be like, why? Why are they so closely related, but one of them can grow on a plate and one of them can't? And surprisingly, they find very few differences between the two genomes. There's definitely, they highlighted here, biotin and asparagine pathways that are inactive. Actually, they're inactive in the other way. The free living one doesn't have them. Oh no, I take that back. They're inactive in the endosymbiote, but they also find one in the opposite way. I think another amino acid is active in the Nucleobacter, but not in the. Oh my god, I can't. Oh, Fastidiobacter. Yeah. So yeah, that's just really confusing. Actually, I guess the authors really expected to see a lot more, because typically when we think about endosymbiotes, we think about gene loss, like either tons of things turn into pseudogenes being expressed, or the genomes even get smaller, like in Rickettsia, right? Genomes are actively getting smaller through time as obligate intracellular parasites in that case. But that's not what they see here. They do see some genome rearrangements, but yeah, there's no sort of smoking gun. There's no smoking gun in the genome to say like this is what went missing, and that means that it can't live in there, can't live out there anymore.
Yeah, it's interesting because I think the main focus would be, I mean, it's almost a good thing that there is a very small part of the genome that's missing, because I think that allows you to hone in on the like the main key points that are driving this endosymbiosis. Mm-hmm. And also the theory is basically that the endosymbiosis at a very early stage of evolution, before all those genes have been lost. So
absolutely,
it's going to be very interesting to figure out exactly what the trigger was. And I think this makes a lot of interesting research coming from that because what I can imagine is like various gene exchange like kind of studies where okay, what genes do we knock out to make the Fastidiosibacter get eaten up by the candidate? I can't remember what the host.
The host is like, it starts with E.
Euploites. Yeah. So trying to get Euploites to eat Fastidiosibacter and trying to get the right genes into. I mean, I think trying to get the genes into the endosymbiote is always going to be a lot harder than doing the free living bacteria. Yep. So that might take a lot longer.
Yeah, I think just maybe somehow swapping in something from the Fastidiobacter into the Nucleobacter and seeing what lets it live on the plate after. I think that definitely.
Yeah. But that is like a whole PhD because again, genetically modifying a bacteria isn't going to be like an easy thing, especially one that is from the wild.
That hasn't been studied before, that hasn't been genetically modified before. Like, yeah.
Exactly. Yeah, it's like, oh yeah, let's just genetically modify it. It's like going up to like, hey, yeah, just make all earthquakes not happen. Easy, job solved.
But yeah, I think as you say, like just having this pair and knowing that the differences are so small between the two organisms gives us this really interesting model system that we could go in and we could discover more about what exactly creates those dependencies. And you know, maybe at the back of my head, part of it is, well, because I think like learning about these close relationships is teaching us about like just so much detail about like what are the requirements for free living versus endosymbiosis, what are the steps along the way, but then also maybe like again, someday we'd like to engineer our own synthetic systems, right, that do certain things. So learning about these principles could help us along there as well. Yeah, I think that's really cool. Also, another thing I'll say is that, you know, as the model, when people talk about genomic reduction, it's a pathogen, right? Rickettsia stuff, those are pathogenic organisms that have genome reduction. So this could also be about the story of like the beginning of pathogen evolution, right? Like where does pathogenesis come from, right? Like what are those links? Maybe. Who knows what you'll find on the journey, but they're all linked, these stories.
Yeah, because I guess pathogenesis is endosymbiosis by another name, where like it's still like organisms trying to live inside of other cells and trying to figure out exactly how that relationship begins. And it's not going to be just one way, because I know that for Listeria, it's obviously because algae were eating Listeria in the wild, and Listeria were like, hell no, let's stop that. And turns out a lot of our immune cells are very similar to algae, so not algae, similar to those amoebae.
No, not Listeria, Legionnaire's disease.
Legionella. Sorry, yeah. Legionella,
yeah, absolutely. There's so many ways to get inside of another cell, and then so many ways that those cells that get inside could find a way to make the most of that situation.
Yeah, and looking at like how it happens, like from a perspective of like symbiotic partners, like is interesting, because how does that start? Does it start like say with the methanogens that are syntrophic? You can I can visualize how that symbiosis evolves in a different way from say a pathogen focused like based symbiosis, because that they're like already partner up even before their cells have like kind of come together. So yeah, this is going to be interesting.
All right.
Speaking of symbiotes, we have Wolbachia-driven, well, not symbionts, but Wolbachia-driven memory loss in parasitic wasp injectatism to enhance horizontal transmission.
So Wolbachia is an endosymbiote. It's an endosymbiote of a lot of invertebrates. Specifically, I think maybe people have heard about Wolbachia can make like mosquitoes sterile or like prevent mosquitoes from breeding in some way.
It can also prevent them from getting malaria and other diseases as well. So there are good bits for the mosquito.
Well, I don't think the mosquito cares about getting malaria.
We never asked.
We never asked. So Wolbachia does this usually through vertical transmission. It goes from generation to generation, and it travels through the eggs. And there's all these interesting ways in which it enforces travel through the eggs. But more and more, it's also being seen that Wolbachia might also transfer horizontally, so between different species. And one of the ways that it's thought to go between species is through this idea of superparasitism. So, like, if a parasitic wasp, let's say, injects its eggs into some poor caterpillar, but then later another wasp comes along and injects their egg into that clutch of eggs, then that's a superparasitic event, right? So it's like a parasite of a parasite is the superparasite relationship.
Yeah. And in an ideal environment, you wouldn't want like the wasps wouldn't want to be superparasites. They want to make sure, because from their perspective, those eggs are already taken, and so they may not. If they see an egg with they see like, so they lay their eggs on the, is it eggs of other creatures or?
Yeah. I think it's inside of. I think it's inside of like a moth larva or something.
Yeah.
Yeah.
grammar. So effectively, like, yeah, so I'm using the word egg a lot because they're laying their eggs on top of an egg.
Yes.
And they're looking at that. And so somebody looking at it go, oh, that's too many eggs. I shouldn't add my own egg to that. Yeah,
but
I have these words in my head.
No, but I think that also it is also a strategy, right? And it depends again on the life cycle of that organism. So it could be that. So in some scenarios, you only want one egg per host because the little larva is going to hatch and eat up the whole host. It needs all of that energy. But then there is a strategy that some insects have where maybe their babies, they don't need to eat the whole host. Right, and so that actually gives them an advantage. So, like, if they do see a wasp sting a place and lay an egg, then they also want to sting there and lay an egg because they know that that's that caterpillar is being chosen. Right, it didn't have the defenses to stop it from being stung. So yeah. Anyways, that's the superparasitism thing, and I guess there's a theory. It's kind of unclear, like, why superparasitism should even happen. Like, is it because the wasps know that they're able to survive in that particular environment, or is it maybe because it's something that the Wolbachia is doing? So in order to encourage horizontal transmission, because you can imagine that if wasps are laying their eggs willy-nilly in all sorts of different wasp egg clutches, then that's a chance for Wolbachia to jump into those other egg clutches, and so that's the hypothesis, that Wolbachia might be somehow getting these wasps to lay their eggs in more places because that helps Wolbachia move around instead of whatever the superparasitism, and the wasps just co-evolved to be like, okay, this is fine. We're just going to make sure that we can make use of the scenarios, right, in which we end up laying our eggs. And so the way that the mechanism they think Wolbachia could force or get a wasp to lay eggs in a whole bunch of different places is maybe it wrecks their memory in some ways, and so they don't really they don't remember, I guess, the cues that they need to look at to choose where to lay their eggs. And so this paper, even though it's about Wolbachia infection, it's actually like wasp behavior mostly. So, like, it's not really about they have Wolbachia infected and uninfected lines of this particular wasp, but the main assay that they're using is training the wasps on either a lemon odor or a peppermint odor, where when they successfully sort of choose the right odor, they're rewarded with like a bunch of eggs. And then they see in all these different conditions either being fed memory inhibitors, so there's some drugs that inhibit memory, or by between the pairwise, infected or uninfected, what changes the behavior of these wasps to associate with the thing they were rewarded for. Yeah, and so, okay, what figure, you're looking at figure 4?
I'm kind of figure 4. Is that illustrating what we're talking about, or is that?
No, so figure 4 is like they look at like a gene expression that has to do with memory. So I think actually let's do figure 1 first, and then
okay, go to figure 1.
Yeah.
Okay, figure 1 is based on memory, right? So
yeah, so figure 1 is based on memory, proportion of choice. So whether or not they choose what they were trained to choose as an incentive. And so in experienced females, so that's the third row down, right? Those guys, they were trained in the system that should make them prefer spending time in the conditioning odor. So you can see that in the light blue bar, that A bar, that one shifted to the conditioning odor side of the graph. But if those same experienced females are infected with Wolbachia, then they look like all the other conditions, which is experienced things given treatments, memory blocking treatments, and also untrained individuals. So this is where they think, like, this is essentially that they're going to hold to say that Wolbachia is doing something to the memory of these wasps. And then if you go down to figure 4, they just kind of they tried to extend that down into the gene expression. Oh, I guess figure 4 is parasitism rate, but like figure 3, they extended into some gene expressions where it's like they can also see that the Wolbachia infection like reduces the gene expression of these 2 genes that are kind of important for memory. Yeah.
Yeah, and so that's an interesting thought because I mean, because now I'm just like, imagining how the flies, because I imagine that memory isn't the only thing that gets affected, but
There's probably all
a key
sorts of
thing
things.
is,
Yeah.
but I think it's something that can be tested. So,
Mhm.
but yeah, I've just got this idea of like flies forgetting and going, did I really lay an egg there?
Yeah.
Oh, I need best try out again.
Yeah, absolutely. I mean, it could just be like it's something about their cognition is being disrupted, right? That impacts memory. So it could just be they can't really make the decision anymore to say like, oh, like this is I don't want to lay my eggs where I already see eggs for some reason, right? Like that's like
Right.
they're less inhibited, right, to lay their eggs in these different places. And maybe like one of the correlates of that is this memory loss. At least I think that's what the authors are getting at here. But yeah, as I said, this paper ended up being more of a like a wasp behavior paper rather than the mechanistic stuff about what the infection is doing. But yeah, a really interesting effect. We already like so many things. Wolbachia is just a fascinating um, uh, bacteria, being an endosymbiote of all these different invertebrates and having such wide-ranging, phenotypically important effects on their hosts. Yeah.
Yeah. So now, next paper is quite an interesting one. The consumption of viruses returns energy to the food chains. So, yeah, this has been hitting the news a fair bit about like the first Virovores discovered. That is, an organism that can subsist only by eating viruses.
Mm-hmm.
So, yeah, I know, like when I was a kid and I first heard about the immune system, I heard about that there were cells that ate like bacteria. Part of me always thought like, hey, is it possible for someone to live off of the bacteria that were infecting them? Which I I credit as my least practical perpetual machine idea. But
I mean, that's kind of like it's almost like that's like tube worms, though, right? Like those tube worms, they just live off their endosymbiotes. So like,
I mean, yeah, but I think those endosymbiotes break down, like take food out. Whereas like
oh, from the
bacteria is eating
environment.
you
Yeah.
while you're eating the bacteria.
Right,
So,
right. That's
uh,
full motion element there.
but yeah, no, I think this is something that people have been thinking about for a while, but no one's actually like gone out there and just proven that this can happen. Because viruses, they're made of protein and DNA and all sorts of things that are delicious, but the thought it's always thought that they're quite negligible
Yeah.
because they're so tiny.
Mm-hmm.
That I mean, theoretically, we're all eating viruses all the time because there are like lots of phages in the environment
Yeah.
that don't infect us, but yeah, just happy nutrient packets for us. Um, but and in this paper they because I think viruses are very common in the sea. There's like tons and tons of them,
Mm-hmm.
uh, and they had the idea that maybe there are algae out there or plankton that could eat virions in water. So they think they found one Halteria that they kind of I think they found it like eating some chloroviruses, and so they just decided to go the extra step and create a pure culture of just chloroviruses and adding these virovores to it, and seeing like what happens to like say plaque propagation. So if we add these organisms to a virus culture, are there going to be less viruses
Yeah.
in the end? And then they went to the extra step of going, okay, let's grow these organisms in just viruses, just surround them in these chloroviruses
Yep.
and see if they can still stay alive after a certain amount of time.
Yep. And they could.
Yeah, and they could. So they found that the Holteria number like increased and the chlorovirus number decreased. So seems like there's eating going on.
Yeah.
And this is something that I think this raises awareness of this kind of, because I think people tend to think about like viruses as like an endpoint in the nutrient web, that
Mm-hmm.
like they kind of just create generic like detritus is what we like
Yeah.
think of them as.
Well, I think
And,
inside of the ocean, right? Like it's particularly in the context of an ocean, like it's dead stuff sinks. And so the idea is that viruses kill stuff and then that stuff sinks and it goes down to the bottom of this ocean where then other nutrient cycles have to make use of what's going down there. But if stuff eats viruses, then it's not just that viruses sink, but also in the virus like exploding out of these bodies that are dying, that also adds food into an area that maybe didn't have food before. Yeah.
So yeah, a really interesting paper to like kind of shift the idea about how we think of like viruses as part of the food chain.
Yes.
In that they're now part of the food chain.
Yeah, like they're actually like a resource that could be eaten. So now, you know, for all these people, like I think that the class of scientists that's looking at this are like sort of very big, zoomed out ecology views of things. And then they're also looking at like they're maybe doing some like large modeling calculations on like carbon cycle or like nutrient cycle in these different ecosystems, and then making predictions. And so now they have, this is like a little experimental evidence to say, hey, did you put this in your big model? Like, did you account for the fact that some things will, like some of the nutrients will also be going down this particular pathway? And so, yeah, that's really interesting from the point of view of, yeah, like a very small observation, but that might have really big implications for a set of science that talks about our ecology and how the world is working.
Yeah. So I'm going to move on to the next paper, which is phage resistance profiling identifies new genes required for biogenesis and modification of the corynebacterial cell envelope.
So I picked up this paper because I think it's sometimes, I think we've said before that we're learning a lot about our immune system and a lot about the host when we study a virus. And I think you can forget too that you learn a lot about bacteria even from studying the viruses of bacteria. And particularly in this model system, they're looking at the corynebacterium cell envelope because it's in the same family as Mycobacterium, which causes a lot of diseases and stuff like that, but it grows a lot faster. And it also isn't as like strict. Like Mycobacterium, like it needs like really strict conditions. A lot of it, if you knock out a certain set of genes, like they die. But corynebacterium are like a little bit more robust in that sense. And so they're using corynebacterium to study this class of bacteria, and then they study it by using phage. And they found that a certain set of phage, like they don't. Some of these bacteria are resistant to phage lysis, or the phage aren't as good at lysing them. And so they were able to track down like one particular gene in the phage, and that could potentially be a receptor for it. And they're like, oh, that seems really interesting. So let's do a whole mutant library of all the different mutations that could happen in corynebacterium that could give them resistance to the phage that they have. And in doing so, they're going to learn about all this different stuff that phage, because like in order to cause an infection, phage have to be able to attach, they have to be able to replicate inside. And yeah, they have to be attached to the surface, they have to be able to get inside. So they're really, all the mutations that they're sorting through, they have some impact on the surface of the corynebacterium. And that is telling us about the surface actually of this whole class of bacteria that includes Mycobacteria. Let's see, what figure did you scroll us down to? Yeah,
I just scrolled you down to figure 3.
Yeah, because I think this is definitely their one of their big findings. Oh well, yeah, it's the arabinogalactan requirements. But I think it's something. Oh yeah, it's actually in figure 4. They do a bunch of different things. They find a lot of things in this screen. But in figure 4, they find like these are pores in the mycobacterial membrane that are required for phage for infection. And then in figure 5, is the thing that they could never have found in Mycobacterium at all, is they actually find that like the mycolic acids are required for this infection. And so in Mycobacterium, if you knock these things out, the bacteria just die. And so this is one of those kind of useful things about using corynebacterium to study Mycobacterium, is that if they got knocked out in corynebacterium, it doesn't kill the corynebacterium, but it stopped the phage from infecting. And so that means that because they know that the phage needs these mycolic acids to enter, they had sort of like found this from the earlier studies, they're able to say like, oh, this gene that we knocked out is important for synthesis of this particular mycolic acid. And yeah, and I think that's something that they wouldn't have been able to find. And this gene has a homolog in Mycobacterium as well. So it's one of those things that like just the particularities of the biological system made it impossible to find this information if you were studying Mycobacterium itself. But because we're studying it in a related object and we're studying it not through like looking at the cell wall and all the things that's being made, but rather just screening through infection by phages, we're able to pull out things that are relevant to the lifestyle of organisms that maybe we do care about in more meaningful ways, like in terms of infection control and so forth. So I guess that was my attempt at very quickly explaining something that's kind of dense and abstract. But the takeaway I feel to get from it is that that like we've said it before, that viruses teach us something about the host. So even in the bacterial sense, that's true. And what's really interesting on the bacterial sense is that, you can also study related bacteria by studying slightly different bacteria, and you get to learn stuff that becomes relevant in different contexts.
Yeah, and I think like understanding how this cell wall is put together is so interesting, because there are so many so many antibiotics out there that work by hitting the cell wall, and also there's like the phage therapy aspects here that could be sort of interesting, but also and also I think like I need to emphasize how much of a pain it is to grow Mycobacteria, because I knew a postdoc who had to work with tuberculosis. It was like 3 months for, I might be exaggerating, but it felt like ages for them to grow the cultures.
Yeah, just to get cultures streaked on a plate, like it's longer than 24 hours in Mycobacterium. I think that already tells you like that's going to take forever to get certain types of work done. But again, if you have like smart choice of a model, you could study things that are relevant to Mycobacterium, but have something that grows a lot faster.
Yeah, so you can get to like your answers much quicker than it can to like iterate through with Mycobacteria itself. So you can like all the basic research stuff that you want to get through, like finding accurate targets. Then you can wait, and then finally, when you've got to your Mycobacteria experience, you can actually go, "Hey, I've actually got some idea of how this will turn out."
Yeah,
and it should be good. And in 3 months, when I take this petri dish out, it's going to show what I wanted to show.
Yep. In some ways I chose this paper because it almost shows that like just by studying bacteria for the sake of bacteria might actually give you interesting stuff that becomes useful in other contexts. Because like the system is it's a phage that infects a Corynebacterium. Like the phage has been bred to infect this Corynebacterium. This Corynebacterium is just like a random fast-growing variant of something that exists out there, right? That doesn't cause any disease, that just lives somewhere in nature, right? But it's related to some other stuff. Like there doesn't seem to be anything intrinsically important about those two things, but by combining them together and like teasing out like the meaning, you find things that you could not have previously found if you chose maybe something more relevant, a more relevant model, let's say, for human disease.
Right.
Okay.
Yeah.
That's all I want to say.
Cool. Our next paper is Artificial Sweeteners Inhibit Multi-Drug Resistant Pathogen Growth and Potentiate Antibiotic Activity.
Yeah, this is a funny paper because, we talk about repurposing drugs that have already been approved for certain, for therapeutic purposes because they're already being through the pipeline. Well, this is almost taking that to the maximum and saying that, like, well, what about, there's other things that also exist in the world already, and I guess there was already evidence that said that it might have some impact on bacterial growth, and that class of molecules is artificial sweeteners. They actually, in the introduction of this paper, it says that it doesn't really get fully digested by our bodies, and so there's actually just a lot of artificial sweetener out there in the world. Contaminating our environments.
I guess that's the point of artificial sweeteners. They're not meant to be They're not meant to give us any calories, so they're not really meant to be digestible. And I can imagine a situation where that would mess with like or other organisms that want to eat like the same kind of sugars.
Oh yeah, oh yeah, exactly. Like it has just enough shape to send off our taste buds, right? To say sweet. So like that must mean it has just enough shape to interact with other things.
Yeah, in the same way that like nucleoside analogs mess with viruses, like sugar analog might mess with other organisms that have a metabolism. So I don't. So because there's all this metabolic enzymes, they'll be like, "Hey, lovely, sweet," and then like, "Go, no, can't metabolize this."
Yeah, so one lens actually that the authors point out in their introduction to look at artificial sweeteners is as environmental contamination, right? Because they're essentially these molecules that are going out into the environment. We don't really know what they do, but they're accumulating because they know very little can break it down. And there's already been some research to show that some of them can promote the growth of E. coli, and then on the other hand, they also showed that it could inhibit the growth of E. coli. So I think the idea is that
it all cancels out.
yeah, I think the idea is that I don't think the field actually has a good idea of what these artificial sweeteners are doing in the environment. And these authors take the approach of the therapeutic lens to say, well, if it is possible for them to inhibit growth, maybe, right? This is like a relatively safe thing for humans to consume that could somehow inhibit the growth of bacteria. And so that's sort of the thought lens that they're starting off with. And so they have a whole panel of artificial sweeteners they're trying, from the sugar alcohols, xylitol and sorbitol, to Splenda, sucralose, or like the classic aspartame, and some variants of that, such as sodium saccharin and ACE-K.
Yeah, I mean, I know, like in my diet drinks, you might occasionally see me drink them on this show.
Yep.
Yeah, it's ACE-K and aspartame, because I was reading the paper and looking at I can see this.
Oh, I know this. This is.
I know this. Regards to my microbiome. Go.
Yeah, I mean, I think that's a really big thing that's coming out. So they do growth tests first to see which ones inhibit growth. And then they take the ones that inhibit growth, and they also test them in another circumstance, inhibiting biofilm production. And they focus on ACE-K because it just has a really marked effect on inhibiting biofilm production.
Yeah, I mean, I need to, because if it affects the growth, it probably should affect the biofilm production, because it's very hard to disentangle biofilm production from like growth as separate things.
I think that they do. Oh yeah, they do. The assay for biofilm production, it doesn't include it. Like it's 24 hours, but they let it grow without the ACE-K before they add it in. So like the biofilm gets established first, and then they add in the artificial sweetener.
Yeah, that's quite nice. I like that as it comes out.
Yeah, so it is something, but you're right, because even in that time that they wait for the ACE-K to do something, like in the condition that doesn't have it, right? There could be it could be like a general metabolism stopper or something like that, right? It's just like if you're not growing, you're dying, like that kind of mentality.
But yeah, I mean. Yeah, and they look at a lot of like interesting things. Like they look at the global gene expression that they focus in on that for ACE-K.
Yeah, I just want to say one more thing about the biofilm thing. I think they also use a concentration that's under the growth inhibitory concentration.
Right. Okay.
Yeah. So like, and I think they will use that concentration as they also do this other stuff and like look at gene expression and things like that. So yeah. I think they do a fair job of disentangling it in that way.
Yeah, that takes a lot of like work to do, so I think that's quite good.
A lot of work, but at the same time, I feel like I mean, it's all just in concentrations, right? And maybe that's one of the wonderful things also about working with these working in this fashion with sort of lots of safe molecules that like you can titrate really easily. They said the only one that was difficult in solubility was aspartame, but they had like full like ability to titrate all the rest. Yeah, like it's just I see a lot of opportunity, like this paper acts a lot on opportunity, like having access to all these artificial sweeteners, having access to a bunch of bacteria and different assays. We're able to collect all of this data and then come to some thought about what might make it important in the environment. And it's not without any basis. They're thinking about its impact, as you say, on the microbiota and so forth. But as they continue their studies, I feel like they start moving towards this idea of like, what if it is a therapeutic?
Yeah. they do test it up against some like notoriously like important bacteria for infections.
Yeah.
So Staph A's, yeah, and Pseudomonas.
Absolutely.
They're both like important for like resistant infections. So showing some kind of effect on those could be really important.
Yeah, Acinetobacter and Pseudomonas aeruginosa are also like classic gram-negative antibiotic-resistant pathogens, and so yeah, showing that it causes membrane. They actually end up showing some microscopy, and it causes like membrane blebs of some sort.
Yeah.
So not really sure why any of this, but I mean they have some hypotheses because they did gene expression, inventories, but they haven't dived down exactly. Instead, they take the path of, again, pushing for this idea that it could be some sort of application by using a biofilm burn wound model on pork skin. And they soak some gauze in their artificial sweetener, and then they show that artificial sweetener soaked gauze is better than just regular gauze in this particular burn wound model.
Yeah, that's quite an interesting thing to look at. I mean, yeah, because I think like there has been a journey of this paper from going like, hey, look at this environmental contaminant to, hey, this could be a treatment.
Yeah, I think that not everyone. It's like this is the classic like final figure stuff, right? Where it's like,
right, oh yeah,
in the attempt to tell a really interesting story, right? Like that has this like narrative that drives towards therapeutics. Like things get slotted into that way. But I mean, to me, it's interesting maybe from diving in deeper into the mechanism of like why exactly does it impact gene expression in all these different ways? Like another phenotype that they pull out, which is both a phenotype but also like a therapeutic application, is like the potentiation of antibiotics, right?
Yes.
Like okay, so it makes antibiotics easier to kill things. And that's probably because we saw the membrane blebbing, right? That the membrane, like having an unstable membrane and an antibiotic is like two stressors that are going to kill something. But like to me, that's like that's just a phenotype actually, not really like it doesn't get closer to the why. Like what about ACE-K is doing this? Like what about how does this help us predict what ACE-K might be doing to things that aren't being directly studied? And I think that to me, the environmental contamination part of the discussion was like really interesting to me from that perspective. And the paper didn't really go in that direction. It went more like oh, this is a potential very safe, a very safe and readily accessible antimicrobial.
Yeah, I mean, it's very easy to market to kids to be like, hey, a medicine that actually tastes good. Yeah, I mean, one of the things I'd be interested in is like the mechanism, because I'm always interested in like what part of the process is this thing attacking? Because I don't think that it's just going to be this molecule that'll be the antibiotic that ends up. It's going to be some iteration of it that's designed to be more druggable and more, because the chemical properties of an artificial sweetener that's designed for aren't necessarily going to be the same as antibiotics. So I imagine that once they to figure out what pathway this is hitting, they'll be like, okay, let's create more molecules that hit that better.
Yeah, absolutely. Yeah, like to me, if this paper is trying to contribute something into the antimicrobial resistance world, then it would be interesting to dive into that mechanism a little bit more, right? Yeah, and if it's and even mechanism is always useful, I guess, from that perspective, right? That it shows us more like the root causes of something that helps us generalize it. But in of itself, I think this paper is quite fascinating, just because, first of all, they tried to do this, right? That they sort of took these little threads of seeing that, oh yeah, like artificial sweetener does affect bacteria in different ways, and then take it essentially through like the standard suite of bacterial assays in order to like figure out whether or not it has clinical significance. It's a preclinical study, to be clear, right? They use pork and
so this is like the earliest stages. There is a lot of like iterations to go through before it's going to be turn into an actual treatment. And as we saw with Molnupiravir, there's no guarantee that it'll be like a a treatment that will be used. There's a lot of like different paths along the way before it gets to that stage.
Totally. I will say though, like in the back of my head as I was reading this, I did think about like, well, there is an interesting like hack though, right? That if it does have a really great antibiotic effect and it's already out there and contaminating the environment, right? This is something that I feel like you could like hack up like a in the zombie apocalypse when you lose access to certain objects, right? But you're like raiding the supermarket and you see like a thing of Splenda, like, you know, take it. If the antibiotics are gone, take the Splenda.
You hear that, Hollywood? There's an opportunity to misinform a generation of people by going, oh yeah, don't worry, put some Splenda on the wound. Salt and Splenda. It'll be it's like the kind of the pee on the jellyfish thing that again has damaged a generation's knowledge of how to deal with jellyfish.
Oh God, yeah. Maybe that is horrible. Also, it's not even Splenda in this case, right? Like it's some. I know sucralose had the same properties. No, sucralose didn't have the properties at all.
Now it would be Ace K, which I think the only thing I know that's in is like Diet Pepsi, and people like people's favorite Pepsi, right? People's favorite soft drink.
All right, let's move to the last paper of today.
Yeah, a GPCR-based yeast biosensor for biomedical, biotechnological, and point-of-use cannabinoid determination. So this is the idea that they're creating a biosensor that's based on G protein coupled receptors, which are these kind of very fast, like acting sensing molecules. And the idea there is that they took some yeast that has like a GPCR sensing pathway, and they swapped out some genes. So they swapped out the main sensor for the human cannabinoid gene. So if something attaches to the cannabinoid gene and activates it, it activates this G protein cell receptor. Yep. I keep you saying different things for the acronym, and it's giving me slight anxiety, but G protein coupled receptor is the right thing to say. And then they train that to like an output reporter. So this paper is them kind of tuning that and then demonstrating what kind of like where it could be useful. So I think the first step is like
GPCR is like they're an incredibly diverse class of signaling molecules. Like they do like I think smell is linked to GPCRs, taste, hormones, immune system stuff is GPCR signaling. So like this class of receptors is like really widespread in eukaryotes. And so I think that that's like really important. They end up using yeast because yeast is a eukaryote. So like it's kind of already like a well-established chassis for this particular set of receptors. But you can imagine with the power of genetic engineering, you could have the diversity of all G protein coupled receptors out there and express them into yeast. So the potential of the platform is actually really quite exciting for me. And they're going to go and examine this through the lens of just this cannabinoid receptor. And specifically, they're linking their synthetic G protein receptor to the yeast like mating hormone, I think, right? It's the the pheromone.
Pheromone, yeah.
Mm-hmm. And so that typically would be like stopping the cells from dividing or something like that. And so essentially they go through that pathway and they make sure that instead of doing the thing that it's supposed to do, it's instead activating the color producing or light producing molecules or fluorescent producing molecules that they need as a reporter to read as output.
Yeah. So I think part of this paper is like them constructing this, putting it into the chassis, and then they select different outputs. So because I think part of them is the thinking about the use cases. So so if they like for some they're looking at like fluorescence, which it might not be useful in some circumstances, but then they look at like color changing and luminescence. So
and the color changing they use is betalain, betanin. And that's the red color in beets. It's actually become very popular in the past like year, I would say. In the past year or 2 years, it's become like the color molecule of choice. And you can, I think, I'm not sure if we talked about it at all on the show, but like, you know, you can understand why. Beets are very red. And they're also that red is very stable. And so it's been an amazing way of doing all sorts of reporters. But the authors note that color production is one of the more is more susceptible to noise, because depending on what sample you use, your sample may impart some color onto the reaction. And so I think their choice later to move into luciferase is made on the basis of trying to reduce noise, because not much in the environment is going to spontaneously create light when adding the correct substrate. But a luciferase reporter would.
Yeah, completely. And they do look at different applications. I think the first thing is like high throughput screening, because they basically got a human cannabinoid receptor. So anything that can hit it can activate it. So they can use it to find new molecules that activate the human cannabinoid receptor, and also new antagonists. So the idea is that if they add like something that they know can activate the CB2 molecule, then they can add like random molecules to see whether any of them block that activity. And I think, they found like, I think, sorry.
Oh, I just want to say, like, this type of screening, it's the basis of drug discovery, essentially.
Yeah.
And so, like having an easy to make cellular reagent that, or a biosensor in this case, that has a relevant output, you get to interrogate the pairwise interaction of its ligand in all sorts of conditions.
Yes, so they found, I think, 2 agonists that promote the CB2 infection, activation, and 2 antagonists that have like never been found before, which again is a good start for a paper about testing this molecule out. Then they did like activity-guided bioprospecting, where they looked in a library of traditional medicine. I think it was Mexican traditional medicine plants they tested through, and they found that and one of the plants did activate this receptor. So the upright prairie coneflower, also known as the Mexican hat, Ratibida columnifera, apparently it activates CB2, and you do with that information what you like.
But I think it's actually really fascinating. Like, even in this example, we're already seeing things like, we know that there's this medicinal application for cannabinoids, and what if we're able to tailor or find other compounds that have these medicinal applications, right? Like, there are other receptors that like complex mixes of cannabinoids activate. If we could engineer those receptors into yeast as well, we could be finding all sorts of things, right? And like start building this knowledge of what these extracts actually do.
Yeah, I think that's really important. And I think another thing that is kind of really good about this, but also really bad for, because there are constantly new like versions of like cannabinoid like coming out to try and evade tests or like get more potency. But so using this as a portable biosensor with the human CB2 receptor means that anything that activates that will come up as a positive. So that will go. So if you're trying to like design a molecule to evade most tests, it's going to be very difficult to make it evade this test without also making it completely inactive.
Yes. So, it's bypassing all that antibody, because like typically the way that we'd be looking for a molecule out there is by an antibody, right? So that has to recognize a certain region, but that's not always like the active site. But then here, because we're saying like receptor binding, and the receptor could ostensibly, it's not always going to be like the GDPCR, GDPCR?
GPCR. We get too many acronyms on this show. I apologize. GPCR, G protein coupled receptor.
No, I think the D came in there because it's like the data.
GDPR, European.
Anyways, having it have that receptor interaction, that might not be biologically relevant receptor activation, but like it certainly is like some receptor activation. I also imagine that in the future, if this becomes a really good platform, that like people will do evolution on the receptors to look for specific things.
Right.
And so point of care is also really great from the biosensor point of view. I mean, again, from the DIY perspective, I really love this, because all you have to do in the betaline one, at least, is grow up some yeast and dilute it in the sample of some sort. And then you can observe it even by eye to get a rough test of positive or negative, which I think is really great. But they go a bit further in the next figure. They actually mill like a a piece of metal that you can put 5 test samples in with a light producing biosensor and clip it onto your mobile phone. Yeah, actually, this I was looking at this. I'm like, this is quantitative. This is quantitative luciferase data from a cell phone. I really love that.
Yeah, really incredible. And I think like because we're looking at this in the case of like, oh, this is a narc bacteria. It's gonna like rat you out to the cops. But
well, it is.
Yeah, it's not. Sorry, it's a fungus. Well, a not so fungus, as I'd say. But yeah, the actual potential of this isn't just for this is a test bed, but it can be used as a sensor for so many other things, or even for drug screening. And I think this is a really interesting technology.
Just because they're using cannabinoids in this particular example, like I think like people should. It's good to open up the imagination, right, and think about all the things GPCR actually sense. And as I said before, smell is governed by a bunch of GPCRs. So we've talked before about dogs being able to distinguish all these different things. Like, I mean, that's all encoded in those genes. Granted, we don't really know which genes encode to what things, and like we're kind of relying on the magic of brains to work for us in the case where we train dogs. But the potential is there, and that's what makes me excited about this platform.
Yeah, we can put dogs out of a job.
Put dogs out of a job. All right. Well, I hope you had fun with us.
All right. Yeah.
Join us in 2 weeks for more cool microbiology news.
Yeah, thank you very much for joining us. We want to remind everyone that while we're very enthusiastic about microbiology and somewhat qualified, it's probable that we didn't get everything right. Science is about thinking critically and asking the right questions. So if you have any questions or corrections, please let us know in the comments.
I totally agree. You can reach out to us over Twitter or Mastodon. We both believe that peer review is a process and that it requires folks to have read the papers in order to make it work. If you think you have something to add or you found something unclear, just let us know.
Yeah, and it's been a pleasure chatting
with you, Danny.
Join us in 2 weeks for more microbiology content. Until then,
Bye.
goodbye.